Good morning, class 8. Welcome back to your biology classroom once again. Today I will start from vegetative propagation by aerial stem. So look at your book. In some plants, such as a strawberry, a slender branch arising from the base of the main stem. मतलब मेन स्टेम जो होता है वहां से एक स्लेंडर ब्रांच जस्ट अराइजेस एंड ग्रोज एंड बेंड्स टू टच द सॉइल मतलब सॉइल छूने के लिए बेंड हो जाता है then it develops roots and buds at the point of contact with soil and develops into a new plant. So look at the picture at first. In case of especially strawberry, a slender branch of stem just arises from the main stem and just touch to soil. After touching the soil, it just produces roots and buds, and then it grows as an identical new plant. In some grasses and mint, the stem grows along the soil, forming roots at intervals and giving rise to a new plant. So look at the picture once again. In case of grasses and mint, from the main stem, there is another stem is arises and just grow as a new identical plant. When the new plant is mature enough, the part of the stem joining the new plant withers away and the plant grows on its own. So last two lines is very important to try to understand. After getting मतलब after getting maturity, plant जब पूरा का पूरा mature हो जाता है, then the parts which actually added with the new plant, they just wither away. मतलब these parts will be disappear. And the plant grow on its own. खुद ब खुद identical plant हिसाब से बढ़ता है. Next, by root. Plants such as sweet potato and dahlia have fleshy tuberous root with adventitious buds. These buds, when detached and planted in soil, can give rise to a new plant. So look at this picture. These are adventitious roots and buds. When these type of roots and buds just getting touching of soil and favorable condition, from them a new plant can arise. So example must. By leaves, some plants such as bryophyllum and kalanchoe produce adventitious buds at the margin of their leaves. When the leaf falls on the moist soil, the buds develop into small plantlets. मतलब in case of bryophyllum and kalanchoe, they produce adventitious buds at the margin of their leaves. Then when the leaves fall on the moist soil, the buds developed into new small plantlets. The fleshy leaves supply food and water to the plantlets as they grow. The plantlets grow into independent plants on the separation from the leaves. Next, vegetative propagation by artificial methods. Vegetative propagation is a faster method of reproduction. Hence, it is used in the gardens and nurseries. So basically, vegetative propagation process हम लोग कहाँ पे apply करते हैं? In the gardens or nurseries. The methods that have been adopted by human beings for vegetative propagation, they are called artificial propagation. So which vegetative propagation basically human used for their uses? This process is also known as artificial propagation. Some of these methods are cutting, grafting, layering, and the tissue culture. So these are the process by which artificial propagation is just happened. They are cutting, grafting, layering, and tissue culture. So what is cutting? In this method, a part of the stem with an axillary bud is cut from a healthy plant. So just इसके नीचे अंडरलाइन करना. स्टेम ऐसा होना चाहिए, हेल्दी तो होगा ही, उसी के साथ with an axillary bud. is cut from healthy plant and planted in a moist soil. Over a period of time, roots develop at the lower ends of the cutting. Also, new leaves develop from the buds at its upper end too from a new plant. मतलब एक्सिलरी स्टेम एक्सिलरी बार्ड के हिसाब से ये स्टेम काटने के बाद जब हम प्लांट करेंगे मॉइस्ट सॉइल में, सो आफ्टर फ्यू डेज फ्रॉम द लोअर एंड ऑफ द स्टेम न्यू root will be rise, then new leaves develop from the tip of the stem and form a new plant. Cutting is extensively used by the gardeners for growing rose, bougainvillea and cacti plants. So this process is basically or extensively used for the gardens. So by this process, we can grow some plants like rose, bougainvillea, cacti plants, etc. Sugarcane and pineapple plants are also grown from stem cuttings. Next, layering. In this method, a branch of plant is bent and a part of it is covered with soil. After few days, root develops from the part under the soil. मतलब जो पार्ट ऑलरेडी अंडर सॉइल के मतलब अंदर कवर्ड है वहां से आफ्टर फ्यू डेज रूट डेवलप्स. The branch is then cut off from the main plant and planted in the soil. It grows as a new plant like lemon, rose, jasmine. Plants are grown in this manner. Next, grafting. It is a method in which two different plants are combined to obtain a plant with superior qualities. इसके नीचे दाग लगाना. For superior qualities, two different plants attached with each other to get a superior plant. A stem cutting called scion, मतलब जिसका stem हम काट रहे हैं that is known as scion, और दूसरा प्लांट जिसके ऊपर हम उसको बिठाएंगे उसको क्या बोलेंगे? stalk. So a stem cutting called scion from one plant is attached and tied to the rooted stem called stalk of another plant. मतलब एक पेड़ से हम विथ एक्चुअली बर्ड जो स्टेम को काट रहा है उसको बोलेंगे स्कियन, और दूसरा जिसके पास ऑलरेडी रूट है ऐसा पेड़ के ऊपर जब उसको हम अटैच करेंगे और टाई अप करेंगे उसको हम क्या बोलेंगे? स्टॉक. After few days, the stalk and the scion merge together due to rapid division of cell and the bulb develops. So these two lines is very important. So after attachment of scion and stalk, they merge together due to rapid cell division. तो कैसे जुड़ता है? Due to rapid cell division and bulb develops. The stalk supplies essential nutrients to the scion. Grafting helps in developing new varieties by combining the features of two plants. It is usually done in the plants such as a mango and rose. In rose, it is done to get flowers of different colors on one plant. So, this process is mainly applied on mango or rose plants. So what is the basic difference between scion and stalk? तो हेल्दी प्लांट जो हम एक प्लांट से काट रहे हैं हेल्दी स्टेम दैट इज स्कियन, और दूसरा अटैच रूट जो प्लांट के साथ हम उसको टाई अप करेंगे उसको हम बोलेंगे क्या? स्टॉक. ये डिफरेंस बहुत अच्छे से पढ़ना. नेक्स्ट, टिश्यू कल्चर. इन दिस मेथड plants are grown artificially in a test tube under laboratory condition. So first line is very important. In the tissue culture process, plants are grown artificially in a test tube under laboratory condition. This technique is also called micropropagation. So tissue culture is also known as micropropagation. For what? In this process, basically, new plants are cultured in a test tube under laboratory supervision. That's why this process is also known as micropropagation. A small piece is cut off from the growing tip of plant. This tissue is grown into a suitable nutrient medium. So after cutting from healthy plants, this tissue basically plant in a suitable nutrient medium in laboratory. It grows into an unorganized mass of cell called callus. So after growing into the test tube, it turned into an organized mass of cell that is known as callus. Small parts of this callus are put in another medium containing hormones to induce cell differentiation and formation of plantlets. सो कैलस से नॉर्मली ये सेल्स को निकालने के बाद ऐसा दूसरा कोई कंटेनर में उसको रखा जाता है जहाँ पे ऑलरेडी हार्मोन प्रोवाइड किया हुआ है. इस दौरान क्या होता है, सेल डिफरेंशिएशन होता है एंड दे फॉर्म इनटू अनदर प्लांटलेट्स. द प्लांटलेट्स आर देन transplanted into soil or pots to grow into mature plants. Since an unlimited number of plants can be produced, this technique is used for the propagation of ornamental plants. So basically tissue culture process is used for the propagation of ornamental plants. So question aa sakta hai, so for ornamental plants which vegetative propagation process is used? So answer will be tissue culture, such as chrysanthemum, asparagus and orchids. So student, hope all of you understood what I've discussed in this video. So for today, up to this portion.